Okay, next speaker is Christine Straub from Germany. Metabolomics and performance data indicate a higher adaptive flexibility in the transition periods in Fleckvieh than in Holstein cows. Let's start your lecture.
Thank you very much for the nice introduction. First of all, I would welcome you all here in Japan. I'm very pleased that I got the opportunity to present my research project on such an international stage. And for that possibility, I would like to thank the organizing committee. As you all know, dairy cows are exposed to dramatic metabolic changes throughout the lactation cycle. In late lactation and the dry period, they are in an anabolic state. And when calving occurs, that situation changes and the metabolism shifts from this anabolic state to a catabolic state. And we refer to that as a negative energy balance, which is a very physiologic situation and can be found among all mammalian species during lactation. So if it's physiological, then there are, of course, adaptive responses in place, because organisms always strive for homeostasis, for a balanced state. And one of the best known adaptive responses is the mobilization of body tissue. And we see that as veterinarians because after calving, cows lose BCAA scores. But then in modern dairy cow management, there are also other factors that are essential in sustaining health and productivity. And those factors include management and environmental conditions. Unfortunately, we have a high incidence of production diseases in early lactation. And key factors in preventing those production diseases are, without doubt, by improving management conditions, for example, feeding staff, housing, and environmental conditions. But part of the problem is also because those adaptive mechanisms fail. And if that happens, then metabolic dysregulation can contribute to a decreased health and productivity. So we have just learned that cows require a highly flexible metabolism to manage those challenges. And a lot of study in the last years have been focused on investigating adaptation strategies in Holstein cows. And when we look on a single farm, as a veterinarian again, then we see cows that can cope with the challenges imposed by the transition period, while others fail. And our initial idea was, well, if there are of course differences on an individual level, there might be also influences that are that derive from the genotype. And therefore, we hypothesized that different dairy breeds manage the challenges imposed by the transition period in different ways. And here I have to emphasize that all cows in our experiment were kept under the same environmental and management conditions. So they were kept on one single farm. And as you can see here, we used two different kinds of breeds. The first breed we used was German Holstein, which is a dairy type breed, primarily selected for milk production. And the other breed we used is a dual-purpose breed, German Fleckvieh, also known as German Simmental, which is valued both for its meat and its milk production traits. And the major criterion to be included in the study was initially a milk yield that exceeded 7,500 kilograms in the previous lactation. But we also looked at various other parameters, production parameters, fertility parameters, as well as data from milk records. And we had the aim to create two groups that were comparable. And then retrospectively, we created subgroups with the aim to further attenuate effects of different condition and production. And because our study had a preliminary character, which means it hasn't done before, and we didn't know, of course, what to expect. We just settled on 2 time points. The first time point was chosen because it reflected a state when cows are in an anabolic state, which was 42 days before expected day of calving. And the second time point was chosen 21 days after calving, so at a time when cows are still in a negative energy balance. And then those samples were subjected to a metabolomic analysis. And you probably ask yourself now, what is metabolomics? I talked earlier about an organism, or the aim of an organism to aim for a balanced state. And in order to achieve that balanced state, to maintain the balanced state, a variety of metabolites are produced at any given time. And as you can see here, a lot of different external and internal factors have a direct impact on the metabolome, on the sum of all metabolites, on the biochemical composition. And therefore, we can kind of say that the metabolome serves as a kind of surrogate for the phenotype itself. Now, therefore, metabolomics is a very useful tool if we analyze biological processes, disease states, or in our case, different adaptation strategies to early lactation. It sounds easy, but I can assure you it is not that simple, because those samples were analyzed by mass spectrometry, and mass spectrometry did identify and quantify over 180 metabolites that could be assigned to the lipid, carbohydrate, and protein metabolism. And when you have a lot of data, it's always important to get an overview. Now, I have to add, I'm not a statistician, but principal component analysis can help you with that. So principal component analysis, put in simple terms, simplifies the complexity of high-dimensional data, but patterns or clusters that exist remain in those data. So what you can see here is a principal component analysis, where each dot represents the metabolome of one cow at one sampling point. So for each cow, 2 points. Now, and if you remember, I mentioned earlier that metabolome is dynamic. It changes over time. And you also remember what I said about principal component analysis retaining patterns. We can see that there's a shift from the left to the right. And because of time limitations, I just would like to demonstrate you a couple of metabolites that differed significantly between our 2 groups. For your understanding, the green columns always highlight the Holstein population, whereas the gray columns highlight the Fleckvieh population. And what you see here are the concentrations of the sum of 3 amino acids. They are leucine, isoleucine, and valine. And they are called branch-chain amino acids. And as you can see here, in the postpartum period at day 21, they differed significantly between the 2 groups. And also have a look at the standard deviation in Fleckvieh, which is very low, which means kind of maybe that Fleckvieh cows are doing pretty much the same thing. And another metabolite that did differ significantly between the 2 groups was a biogenic amine, which is called 2-aminoadipic acid. Here you can see that Holstein cows displayed significantly higher levels than Fleckvieh cows 21 days after calving. And you ask the next question probably now, what are the roles of those metabolites? And to be honest, the underlying mechanisms are poorly understood. So if you have spare time, you might be able to dedicate some of your time in unraveling these mysteries. But several studies, and some published recently, suggest that elevated levels of branch-chain amino acids result in the activation of transcription factors in immune cells, which in turn result in the production of pro-inflammatory cytokines. And the same, unfortunately, is true for the metabolism, where elevated levels of branched-chain amino acids are associated with insulin-resistant states. But how these elevated levels occur, or how they contribute, how they lead to insulin resistance, are unfortunately not fully understood. And the same is true for 2-amino adipic acid, which is believed to be a modulator of glucose homeostasis. So based on those results, it's probably quite tricky to say that there's a higher adaptive flexibility in Fleckvieh cows than in Holstein cows, and I agree with you. But there are other parameters which have been used in the past to describe energetic imbalances. And the first one is beta-hydroxybutyrate. And what you can see here is that in 21 days after calving, the concentration of beta-hydroxybutyrate was higher in Holstein than in Fleckvieh cows. And the mean concentration of beta-hydroxybutyrate in Holstein was even above the threshold indicated for succinate ketosis, which is in this case 1 millimole per liter, despite similar milk yields. And another parameter that can be used to kind of assess ongoing mobilization of body reserves is by calculating daily body weight changes. And we did that, and you see that there's a significant difference between daily body weight changes between Holstein and Fleckvieh cows. Here in this graph, positive values indicate body weight loss, while negative values indicate body weight gain. So to sum up, Holstein cows in our study displayed higher beta-hydroxybutyrate levels in the postpartum period than Fleckvieh cows. The body mass loss was ongoing in Holstein cows, while this effect was not apparent in Fleckvieh cows. And lastly, with this analysis method, we identified 2 metabolites that are, at least in humans and rodent models, indicators or markers for poor metabolic health or not good adaptability. And therefore, we conclude that there's a higher adaptive flexibility in Fleckvieh cows than in Holstein cows. I would like to take the chance to thank all my supervisors and co-workers for their help, advice, and support. I know that there are a lot of unanswered questions, but I mean, that's the core of research. That's why we are all here. And to take the words of Albert Einstein, if we knew what it was we were doing, it wouldn't be called research, would it? And with that, I thank you very much for your time and your attention.